Just play that out a bit further. What does it mean to be subordinate in a kindergarten context? You've done some very interesting observational studies, haven't you? And then measuring salivary cortisol, so cortisol levels in the saliva of those little kids. But what does it mean to be subordinate in a kindy hierarchy?
Yeah. Any kindergarten teacher can tell you that within 2 weeks of 20 kids coming together in a classroom, there'll be a pecking order that is linear and transitive, with kids at the top and kids at the bottom. There are children who imitate each other, who defer to one child versus another, who automatically get to go to the head of the line instead of the back of the line. And this is something, if you think about it, we're all basically familiar with, not only from our adult experiences, but from our childhood experiences. And it turns out that this ranking of children has certain things in common with the ranking that occurs within societies. And so we've been doing observational studies looking to see whether there are differences in things like stress reactivity, if there are differences in sort of preclinical psychiatric conditions, behavior problems, and differences in physical health problems as well.
So, Tom, how significant are the changes in cortical activity in those children quantified as being at the bottom of the rank? And how long do those changes last for? Because this is key, isn't it? I mean, the stress system is a good system. Cortisol is a good hormone, but it can be cast as a bad hormone too.
Yeah. We show that there are differences in cortisol responses to a set of standardized challenges when we bring kids into the lab and the child is a subordinate versus a dominant child. But there are other investigators, Sonia Lupian at McGill University is an example, and this is Bruce McEwen's work as well, showing that even basal levels of salivary cortisol are higher among kids who are from lower SES families. There may be changes over time, and kids may, sort themselves out in different ways as they go into adolescence and into young adulthood. But we think that there is a tendency, at least at the group level, for kids who start out high in HPA reactivity and basal secretion to stay there.
Now, this is very significant because it has all sorts of effects down the track, doesn't it? Tell us about the dental carey study that you did.
Yeah. We realized at some point in our study of kindergarten children that one of the effects of cortisol over the longer term, when it's constantly secreted high, such as in adults with Cushing's syndrome, is that it causes a depletion of calcium in calcified tissues like bones. People with Cushing's disease get osteopenia and osteoporosis. We weren't going to study the bones of these kindergarten children because they're presumably all fine, but we did realize that within the next year they were all going to have teeth falling out, which is another calcified tissue. Mm-hmm. So
you became the tooth fairy.
It was called the Tooth Fairy Project, and we paid kids $10 a tooth. By the way, as a little aside, I think we paid them a little bit too much because when one of the teeth came back from the dental school where we sent them, and they said, "This is not a human tooth, this is a dog's tooth." So somewhere in Berkeley there's a dog missing a tooth. But we began studying these exfoliated teeth as a way of getting at potential long-term effects of hypercortisol secretion. And we have in fact found that the kids who were more reactive in the HPA axis in fact had thinner and less dense enamel within the teeth that had been exfoliated. So we think that may be part of the key to why subordinate kids get more caries, is because they have greater cortisol secretion, and this comes out in the saliva and may change the enamel density and thickness.
And as we've heard, higher resting levels of cortisol are also linked with elevated markers of inflammatory processes in the body, which Tom Boyce says have also been recorded in disadvantaged kids, and which point towards higher rates of cardiovascular disease, chronic pulmonary disease and mood disorders in adulthood. This is All in the Mind on ABC Radio National, abc.net.au/RN, going global on Radio Australia and as podcast. I'm Natasha Mitchell. Now, what about the brain? Bruce McEwen argues It's been neglected as the organ of stress. And his work is revealing how significantly it can be remodelled. You'll remember leading neuroscientist Fred Gage on the show a few weeks back talking about our plastic organ of memory called the hippocampus deep inside the brain. Turns out it's very receptive to cortisol. In fact, the stress hormone helps us remember better, but too much of a good thing. And in rats at least,
we find that certain neurons in the hippocampus, when under chronic stress, they're dendrites, these big trees that are the receiving points for information from other nerve cells, they shrink.
So they're like, around every brain cell are these sort of sprouting limbs, if you like, and dendrites are what bring the signals in.
Right,
and some people will have heard that axons are what take the signals out. So what happens to those branching dendritic structures?
In the hippocampus, they shrink, and there are fewer connections. But then a colleague in India, Shona Chatterjee, discovered that under the very same conditions of stress, neurons in the amygdala were actually growing and forming new connections, which is an organ of fear and, if it's overactive, of anxiety. And the animal was becoming more anxious. Now there's another brain area called the prefrontal cortex.
Which is at the front of our brain. It's our executive brain in many senses. It helps us plan and decide, prioritise, that sort of thing.
And it governs our emotions. It keeps the amygdala in check. And it helps to decide, along with the hippocampus, which is very important for remembering where you are, the context of what is happening. It helps to decide whether these situations are worth paying attention to, and it keeps the system from overreacting. And then with our collaborator, John Morrison, we discovered that the prefrontal cortex, there's a part of the prefrontal cortex in which these neurons are shrinking and losing, the dendrites are shrinking and they're losing connections. So they're losing very important input. It's a reversible process. If you stop the stress, it will grow back.
Now, what does that mean? What impact does that have on a person's behaviour, or in this case, a rat's behaviour?
Well, what we found is that when the animal, the rat, is challenged with a complex task in which it has to shift the meaning of cues that predict where a food reward is. If the task is difficult, having either a lesion of the prefrontal cortex, which other people did, or chronic stress, reduces mental flexibility. Our ability to shift is not totally gone, it's just much slower and less efficient.
Which sort of makes sense. If there are fewer sort of dendritic arms receiving signals, those cells aren't as receptive to the signals that they receive, I guess.
And then if we just stop for a minute and talk about translation, the graduate student who did this work, Connor Liston with us, is also a medical student. And he studied some of his fellow medical students studying for the board exam, the big exam.
Very stressful situation.
And he used something called the perceived stress scale, which actually asks you questions about how much of control of your life you are in and what things have happened that are causing you to be stressed out. What he also did with a colleague is to develop a human task, which was very much like what he did with the rats. And they used functional brain imaging to define a circuit that was activated by this task. They could observe this circuit in these stressed students, and they found that the more stressed out, the less efficient was the circuit, and they also showed an impairment on this behavioral task. Now, what they couldn't do was to look at the detail of the synaptic connections, but we believe that what we learned from the rat is translatable to the human. Now, the good news is that when these students had taken the test and had a month off, all these effects disappeared.
So they're reversible?
Yes. So that emphasized the reversibility in the normal, healthy brain. Now we're beginning to suspect that as the brain ages, it may lose some of this resilience.
Some of the work that we've been doing with Mark Kishiyama and Bob Knight, comparing low and high socioeconomic status kids, shows that there are systematic differences in the function of various circuitries within the brain. Mark's work has shown, for example, that there are differences in prefrontal cortical activation, the part of the brain that's out in front behind the forehead. It appears that children from low socioeconomic status environments have greater vigilance, but at the expense of the ability to filter out kind of incoming stimuli. And if you think about it, it makes sense that kids growing up in poverty deal with far more in the way of threats, severe stressors, things that happen in their environment that they have to be vigilant toward in order to protect themselves and often to survive. But it comes at the expense of developing the ability that also is located within the prefrontal cortex to discriminate between stimuli that are important and those that can be ignored.